the Basque Country. I'm from here, Alexandria University, and this is the place where I did my postdoc last year, where we did a preliminary work about the using non-viral vectors to transfect retinal epithelial cells. The group I joined actually did previous work using these non-viral vectors, lipid-based. They started using solid lipid nanoparticles, and they used polymers as well. And finally, they developed, one of the group who is fortunately with us today, Mohammed, has developed a new, relatively new technique that is being used in drug delivery, but not very famous in gene delivery, which is the niosomes. I will talk to you about a little bit about this approach a few minutes later. These are the authors, and as I said, it's a collaborative work between Alexandria University and the University of the Basque Country, Spain. This is my agenda. Talk briefly about the non-viral vectors, how the idea came about, and the aim of the work, methodology we used, the results we got, and final conclusions. To start with the background, that as you know, that RB cells are involved in the pathogenesis of myriad retinal disorders. Thereafter, they are key target for several gene-based therapeutics. Non-viral gene vectors are thought to be immunologically inert and potentially safe than the well-known and very famous viral vectors used in vivo. Among the different types, we would focused on the lipid-based gene vectors. And among the lipids, we focused on the original cationic lipid that was generated by Fligner et al. A few decades ago, and it has been used in several trials, in vivo trials, trying to deliver genes via systemic route. It has been extensively used then, since developed, and also had been modified, and because it's a synthetic lipid that has a cationic, it's a cationic, so it's positively charged. It has been modified since it has been developed by Fligner et al. This is the structure. As you can see here, we have quaternary ammonium compound in the head, so it's positively charged, and this is the hydrophobic tail. The DOTMA-based liposomes were formulated previously for systemic gene delivery, but to date, it has not been formulated as a niosomal gene delivery vector. So what is the difference between a liposome and niosome? Simply, this is the structure of both. We have a bilayer formed of lipids. The hydrophilic heads are towards the aqueous medium, either inside or outside, and the hydrophobic tails are escaping this hydrophilicity in making this bilayer vesicle. If this wall is formed of a phospholipid, so this would be a liposome, it's formed of basically of surfactant. Non-ionic surfactant would be a niosome. Niosomes are non-ionic surfactant-based vesicles formed of organization of the surfactants, micromolecules as a bilayer, as you can see in here. As a novel gene delivery system, niosomes have received much attention by time for being osmotically active and more chemically stable compared to the liposomes. Besides, when it comes to easy handling and low toxicity, they are considered advantageous over the alternatives liposome. Therefore, we thought these niosomes have a great potential in delivering our genes. The surfactants, we have lots of surfactants to try each and every one of these, and we have tried several, but regarding polysorbate 60, it's very famously used in drug delivery, but not in gene delivery. So we started to try using this surfactant. As you can see, the hydrophilic head and hydrophobic tail would be arranged in a niosome. Usually, liposomes or niosomes, they use a helper lipid to aid and add value to the formulation or enhance the formulation in a way or another in the form of the physicochemical characteristics or even in the function. So we started searching a great library of natural lipids to search for what would be suitable for treatment or delivering genes into the RP cells. We used a lot. Recently, we published, our group published a paper using squalene, a natural tetraterpene, and amongst the working ones was the lycopene. Lycopene, as you know, is the bright red carotenoid pigment found in the tomatoes and red fruits and vegetables. This is the structure of lycopene. As you can see, it's totally hydrophobic structure because it's symmetrical tetraterpene assembled as 8 isoprene units that are entirely formed of hydrogen and carbon. So it's a very hydrophobic structure. I would mention in a minute why. So the aim of this work was to design a novel niosomal vector, but we ended using comparing two actually. One we used polysurpase 60 and DOTMA, the cationic lipid, in the presence of lycopene, and compared to the one without lycopene to know if lycopene really added something to the formulation or not. So because it was, to our knowledge, the first time that lycopene could be used as a natural lipid in formulating liposomes or even iosomes. And we speculated that the comparison would be of great importance between the two vectors to elucidate the potential desirable effect of lycopene in the current formulation. Our methods started with the elaboration of the niosomes. They were elaborated by Muhammad, as I said, and he used a very commonly, routinely used technique, reverse phase evaporation technique, in which he dissolved polysurpase 60 and DOTMA plus or minus lycopene in dichloromethane, which is a organic solvent. This is the oil phase, and we're sonicated with the 5 milliliters of deionized water. After sonication, our niosomes were formed, and then dichloromethane was left to evaporate under, for two hours, this after optimization. And so we got two formulations. We've abbreviated them as D for DOTMA, P60 for polysurpate 60, and DP60, L for lycopene. This is the DP60, and this is the formulation with lycopene, which we thought that, as you see in here, we can see a blue and black color. The majority is for blue, which is polysurpate 60, the majority of the formulation, because it was formed of a molar ratio of 4 polysurpate 60 to 1 DOTMA, very low amount, to 0.5, which is the lycopene. So in here, no lycopene. In here, lycopene is present, and we suppose that it is present in the hydrophobic part due to the hydrophobicity of its structure. Then we formed the nioplexes simply by complexing our niosomes with the GFP coding plasmid, so that plasmid would be complexed with the niosomes according to, we used various NP ratios. NP means cationic lipid to DNA ratios. So we used different weight-to-weight ratios to know which ratio would be actually working if something would work. And then we characterize these nioplexes regarding their size, polydispersity index, and zeta potential using the Malvern zetasizer. And we characterize them morphologically using transmission electron microscope. And finally, we characterize the ability to complex DNA release and protect DNA against DNase enzymes using gel electrophoresis. After that, we did our transfixion study using RP19 cells in vitro in 24 well plates. And after 72 hours, we detected the peak of detection of green fluorescence protein detected by the microscope, fluorescence microscope, and also were detected using quantitatively using a flow cytometer after staining the cells with pyridium iodide to detect if any toxicity or induced apoptosis were induced by our formulations. And according to what we saw, what I'll tell you later, in our transfiction, we were motivated to see the uptake ability of our cells towards the niosomes or nioplexes, and also their trafficking mechanism, the proposed trafficking or internalization mechanism. We used also flow cytometer and confocal microscope to detect so. And I classify my results according to two main criteria: the characterization of nioplexes and then the results of the in vitro experiment. Regarding the size, the charge, and polydispersity index, as you can see here, the niosome, not the nioplex, as you can see, the size is very, very small, fair polydispersity index, and the zeta potential is highly positive, which is believed to be a good value for niosomes, because when you have high zeta potential in positive territory and at this zone, 33, 45, so it is supposed to be a very electrostatically stable formulation. And regarding the size of the nioplexes, you can see that the size is fluctuating. The dark ones are with lycopene, the light ones are without lycopene, regarding the bars. And we have fluctuation on both formulations regarding the NP ratios we used. At different NP ratios, we have fluctuations, but at the end, we can say that it's fluctuating from 150 to almost 100 nanometers, which is believed to be a good size for a non-viral vector. However, I have to say that there are lots of claims about the most appropriate size, and there is no consensus yet about the most appropriate size for the vector. Regarding the zeta potential, you can see that this is the without lycopene. It's very higher compared to the addition of lycopene. But again, we can see that both are above 20, which is a good regarding the stability of both formulations. Regarding the morphology, as you can see, the sizes, no definite sizes, so also the morphology is no difference, major difference we could see. Maybe if we use more sophisticated tools like Crowne or more sophisticated microscopes, maybe we could detect morphological difference, but at this point we didn't. All we can see in here that we have dispersed nioplexes without any aggregates, which is advantageous for our formulation. Regarding the gel retardation assay, as I told you, we did that to make sure that our formulation was able to capture, release, and protect against DNAs. This is the control, and every three is formed of without anything, the first one. The second cell would be with SDS detergent to detect the ability to release, and the last one, the third, which is 3, 6, 9, 12, would be to protect against DNAs. What I can show you here that if you look at this part, even with 22:1 NP ratio, you can see that the supercoiled and the open circular are both evident in both these cells. So as you can see also in 18:1, 12:1, 6:1, and all of them are able to capture, release, and protect DNA. The same is in lycopene containing formulation, almost the same, so no difference. So we tried both formulations for in vitro experiment to see if they would work in vitro or not. To our surprise, as you saw before, no great difference in the physicochemical characteristic between both formulations, but their work was very different, because the light ones without lycopene, as you can see, fluctuating from 2 to 4% of transfixion, which is very, very low percentage of transfixion. And while the lycopene containing formulation actually gradually increasing till they reach their peak at 18:1 NP ratio. We used to compare with lipofectamine, which is known to be a good acting transfixion agent that is commercially available. So we can see with our best NP ratio, we got transfixion of almost 35%, very close to the 40% of lipofectamine 2000. But regarding the viability, we had significantly higher viability compared to the viability of lipofectamine, except for the highest NP ratio we got. So our best concentration was 18:1 using the lycopene containing formulation, and it has a very, very high viability above 96 or 97% viability, which is very high. I forgot to tell you that the negative control didn't show any transfixion, which is not shown here. With a microscope, it shows the same thing. The transfixion gradually increasing, as you can see, and also shows that how much cells look very healthy, starting from the very low NP ratios to the high NP ratios, no great difference in the viability. As you saw before, the formulation looked like the same but acted totally different. So we started to think what made this difference. Why lycopene containing formulation would act very, very successfully and the other one did not act at all. We thought maybe the uptake could play a role. So we did an uptake study ranging from 2 to 4 hours, and we saw that opposite to what we thought, that the lycopene containing formulation had significantly lower uptake than the non-lycopene containing formulations. And with confocal microscope, we saw the same thing. After 2 hours, after 4 hours, you can see accumulating FITC containing or FITC lipid neoplexes compared to the lycopene containing ones. And this is very bizarre because we thought that the formulation that works the best is uptaken the best, but this is not the case. It's not uptaken the best at all. And this could be related to the difference in the zeta potential between both formulations, because the non-lycopene containing formulation has higher zeta potential compared to the lycopene containing formulation. Also, we did internalization assays to know what was the mechanism behind the uptake, because it seems to be something what's beyond the success is something that was not happening at the door of the cells, but it's happening in the cytoplasm and in the nucleus. So we did internalization mechanism using cholera toxin for the caveoli mediated endocytosis, transferrin to see that clathrin mediated endocytosis, and dextran to detect the micropinocytosis. As you can see, at M.6, which was our cutoff level of co-localization between signals, we saw that in non-lycopene containing formulation, it co-localizes with the 3 markers. So it's uptaken by the 3 markers, and this could be of role that may explain a little bit how the uptake is higher and higher, because it used 3 different mechanisms of uptake. However, on the other side, the lycopene containing formulation co-localized with caveoli and micropinocytosis, but not with clathrin mediated endocytosis, which we think it could be the advantageous point behind the lycopene containing formulation, because it was not co-localizing with the clathrin mediated endocytosis, which is known to end by lysosomal degradation. So it could be escaping this lysosomal part. Another explanation, because using lycopene in drug delivery and gene delivery is not an easy thing to search the literature for. So we actually find ourselves in need to search for everything ourselves. It is thought that lycopene could be internalized to the nucleus easily, It has nuclear receptors, so its nuclear localization could be easier, and this might explain, but this I know needs more work to do before we move to the next step, which is an in vivo experiment. By now, we can conclude that by adding lycopene, we achieved significant improvement in transfection of hard transfect human RP cells with excellent viability, which is very important in non-viral vectors because maybe you need to re-inject the patient again or the animal again. So the issue of viability is very important. The potency of a lipid-based formulation depends on both cellular uptake and intracellular trafficking processes. And the cellular uptake of non-viral gene vector does not closely correlate with the efficiency of transgene expression. So we had 80% uptake after 4 hours and 4% transfection in the DP60. However, we had 34 uptake after 4 hours in lycopene formulation and 34% of transfection. It seems to be every cell that was able to uptake the vector was transfected by the end. So it has to be something that happened inside. Maybe we have to go through signaling pathways assays. We have to go through more sophisticated characterization of the arrangement of the lycopene to know better about the cause behind this. So this would be our next step indeed. I would like to thank my collaborator Mohammed, Gustavo Porras, John Zarate, and my supervisor, Professor Jose Luis Pedroth, and thank you very much for your attention.
Any questions? What made you use the lycopene in the first place?
It was not the exact selection of lycopene. We searched about a great library of components. How many, Mohammed? 20 or 25? Yeah, about 20 natural, neutral lipids to be used instead of the routinely used cholesterol and dopey that are used already. So we started to search for new materials to be added that are natural and of neutral charge. Lycopene in the eye is very appealing because it has been reported to be involved in the prevention of oxidative stress of retinal pigment epithelial cells in published work. It is supposed to be acting in protective role against AMD. So it was appealing to try lycopene, but it is not the only one working in our formulations. It's just one of them. So lots of them had worked finally, others didn't actually. One of them is the already published paper of squalene. So that was the reason, the sense behind.
Any other questions?
Have you used lycopene with liposomes, particularly cationic liposomes?
No, we didn't. We didn't use liposomes at all. We used niosomes. During my work, during my collaboration with them, we used niosomes only, not liposomes.
Why did you choose liposuctin 2000? Because now there are more efficient transfection agents than this one. I mean, even liposuctin 3000, Fusion, Attractan, and so on.
Yes, actually, you are right. We have a variety of commercially available agents or vectors to be compared to. You cannot compare all of them cost-wise, but the reason was that it was routinely used in several cell lines, and it was working fine in RP cells. It get transfection RP cells 40% or 42%. So we prefer to compare to a better working agent in our special cells.
Is there any literature on the action of lycopene in endosomes?
No, I didn't. I searched a lot about that. I hoped I could find an explanation of what was happening. But the only thing I can say that it is supposed to be decreasing a little bit the arrangement of the or perturbing or disturbing the lipid bilayer structure. if it did so, why it didn't happen at the internalization at the beginning? This is very bizarre, but it seems to be it worked like this in a way or another later on in the endosome. So I think it's all about the escaping the endosomes and internalization into the nucleus. The exact mechanism, maybe later I can find, hopefully.
And there's, you may have shown this, I'm sorry, but there's no difference in plasma copy number between with and without lycopene, like there's more DNA.
We didn't do quantitative assay for copies of the plasmid. All we did was the percentage of the cell that already have taken the FITS labeled plasmid. That was the point.
Did you do any functional studies to look at the effect of lipo?
No, not yet. Not yet.
So basically, it's just the method of delivery.
Yeah, at the moment, yes. We have to. That was our first step, to set a working vector, and then to study its first of all its mechanism of action, and then it's maybe it has an effect on other facets of retinal pigment epithelial cells, because lycopene especially is thought to be working somehow specifically on RP cells.
Yeah, but you didn't do any
Not yet.
analysis
Not
to see.
not yet.
Okay, thank you so much.
You're welcome.
And